Hello, everyone, and welcome to Advancing the Practice of Wastewater Surveillance, which is the sixth seminar of the Surveillance Advances seminar series, co-hosted by the Public Health Agency of Canada and the National Collaborating Centre for Infectious Diseases, or NCCID. My name is Signe Berger, and I'm a project manager at NCCID. I will be your moderator today, along with Jackie Roberge-Dao, who is a senior policy analyst in the Data, Surveillance, and Foresight branch of the Public Health Agency of Canada. NCCID is funded by the Public Health Agency of Canada to provide knowledge and evidence for use in public health planning, programs, and policies related to infectious diseases. We are incredibly pleased to be co-hosting this series with the agency. I would like to acknowledge that NCCID is hosted by the University of Manitoba on the original lands of the Anishinaabe, Cree, Oji-Cree, Dakota, and Dene peoples, and on the homeland of the Métis Nation. At NCCID, we strive to honor the lands and their original caretakers in our work. We acknowledge that we are on Treaty 1 land. We recognize that this and other treaties have been implemented as part of the process of colonization intended to benefit some while harming others. We are committed to working with our partners towards reconciliation. Before we dive into today's seminar, there are a few housekeeping items I would like to mention. A recording of this seminar and presentation slides will be available shortly after the seminar on the NCCID website, nccid.ca, under webcasts. The chat box for participants has been disabled for this session. The hosts will use the chat box to communicate additional information with participants. If you have technical problems with Zoom during the seminar, please email us at nccid@umanitoba.ca. Please send us topic-related questions through the Q&A tab, and we will try to address as many as possible in the discussion period after the presentation. You can like any of the questions to push them up in priority. We would also like to note that Surveillance Advances is a Section 1 self-approved group learning activity as defined by the Maintenance of Certification Program of the Royal College of Physicians and Surgeons of Canada. The seminar series is also approved by the Council of Professional Experience for professional development hours for members of the Canadian Institute of Public Health Inspectors. If you require a letter of participation, please look out for the survey that will be shared after the seminar.
Merci, Signe. Donc, bienvenue encore à tout le monde et merci d'être avec nous aujourd'hui. Welcome and thank you for joining us today. Une note pour dire que le séminaire d'aujourd'hui se déroulera en anglais et en français. Donc, j'aimerais commencer par reconnaître que je vis et travaille en territoire autochtone. Je reconnais la nation Kanien'kehá:ka comme gardienne des terres et des eaux sur lesquelles je suis aujourd'hui. Tiohtià:ke, Montréal, est historiquement connu comme un lieu de rassemblement pour de nombreuses Premières Nations et aujourd'hui une population autochtone diversifiée ainsi que d'autres peuples qui y résident. C'est dans le respect des liens avec le passé, le présent et l'avenir que nous reconnaissons les relations continues entre les peuples autochtones et d'autres personnes de la communauté montréalaise. Aujourd'hui, vous nous rejoignez d'un bout à l'autre du pays et les terres d'où vous nous rejoignez représentent les expériences vécues par de nombreux peuples autochtones qui ont tous leur propre histoire en matière de santé. Nous souhaitons reconnaître que la pratique de la surveillance de la santé publique peut susciter des inquiétudes chez certaines populations autochtones et d'autres communautés. Pour les professionnels de la santé publique, la surveillance évoque la notion d'épidémiologie, qui consiste à comprendre la distribution et les schémas des déterminants de la santé et des maladies dans les populations. Mais pour d'autres, la surveillance peut avoir des connotations négatives en matière de sécurité, de respect de la vie privée et de contrôle. Donc, dans le cadre de cette série de séminaires, nous tenons à préciser que nous utilisons le mot « surveillance » pour désigner l'évaluation des menaces de la santé publique et la réponse à ces menaces, ce qui constitue une fonction essentielle de nos systèmes de santé publique. Aujourd'hui, nous vous invitons à poser vos questions dans la langue officielle de votre choix en utilisant la fonction questions-réponses tout au long de la présentation. So throughout the presentation, we encourage you to ask your questions in the Q&A box in English or French. Donc, c'est avec grand enthousiasme que nous allons vous présenter nos invités pour le séminaire d'aujourd'hui. Donc, pour commencer, Dr. Caroline Huot est médecin spécialisé en santé publique et son expertise porte sur les risques microbiologiques dans l'environnement. Elle travaille à l'Institut national de santé publique du Québec en santé environnementale dans les domaines de l'eau, de l'air, de la surveillance et des urgences. Elle est également professeure de clinique à l'Université Laval.
A microbiologist by training, Christine Jobin's experience in public health focuses on the wastewater surveillance program at INSPQ. She also contributes to other surveillance activities at INSPQ as a scientific advisor. Further, Christine has experience in the medical devices industry, particularly in the development of rapid diagnostic tests for infectious diseases. Dr. Inès Levad is a clinical specialist in medical biology with expertise in bioinformatics, next-generation sequencing, microbial genomics, and molecular epidemiology. Dr. Levad is currently in charge of the surveillance and genomics of SARS-CoV-2 program in Quebec. Welcome, Dr. Huot, Christine, and Dr. Levad. We are happy to have you all join us today, and we are excited to learn about your experience and knowledge in wastewater surveillance. The floor is yours, Dr. Huot.
Actually, I will start the presentation. So thank you for this introduction, and thank you all for joining. So today we will talk about the Quebec wastewater surveillance program that was put in place for the COVID-19 in the province. I will speak in English. All our slides are in English. Caroline will speak in French and Ines in English, but don't hesitate to ask your question in language of your choice. Next slide. So we have no conflict of interest to declare. Next slide. The objective for today's presentation is to learn on the current research state of the wastewater surveillance and to gain insight into the validity and feasibility of the wastewater surveillance, more specifically for COVID-19, and also to explore the advantages of sequencing of SARS-CoV-2 in wastewater. Next slide. I will start the presentation with a brief overview of the wastewater principle and the Quebec wastewater surveillance program. Then Caroline will talk about a literature review that was done at INSPQ on the feasibility and utility of wastewater surveillance for COVID-19. Then I will talk about the program evaluation that was performed with the data gathered during the first year of the program. And then Ines will address the genomic portion of the program for circulation and diversity through community wastewater sequencing in the province. Next slide, please. Next slide. So to give a brief overview of the wastewater surveillance principle, first it starts with an individual in a community that get infected by a virus. Wastewater has the advantage of catching any individual, whether or not they have symptoms of the disease, and also it is independent of any screening policies that are put in place. The infected individual will excrete the virus in different routes, including stool, which will lead to the detection of the virus in the wastewater. So then wastewater can be sampled in different places, including the treatment plant, which is at the end of the sewage network. And then once sampling is done, the virus, so next slide for the sampling, sampling can be done. The virus particle from the wastewater will be concentrated and extracted. And usually the PCR method is used for detection and quantification of a specific gene on the targeted pathogen. The lab data will then be interpreted and standardized, and we can perform a quantitative relationship against clinical data to follow trends or any correlation with the clinical data. Next slide. This is to give a brief overview of the Quebec surveillance program that was launched in March 2022. So for the first year of the program, we had up to 19 sites spread in 14 different cities, which covered up to 48% of the province population. The sampling frequency for the first year of the program varied among sites, ranging from 3 days per week to 7 days per week. And at the launch of the program, it was a partnership between the public lab and academic lab. So public lab were from the public health laboratory, the LSPQ, and also collaboration with the Ministry of Environment. And we also had the collaboration of the academic, which was the research group Centro. Centro had deployed a pilot project prior to this program that captured many sites in the province. Then in March 2023, we had a reduction of the program, which went down to 4 cities, 1 site per cities. And at that point, the program became only public, so no more collaboration of the academic for the sampling and analyzing of the samples. And since July 2023, we have 2 sites, one in Quebec and one in Montreal, and this is the current state of the program. We have sampling 3 days per week for both sites that are in the program. And yeah, we still with the public partners, and we are testing for SARS-CoV-2 only. So this is it for the brief overview of the program. I now pass the floor to Caroline for the literature review.
Merci, Christine. As Christine said, I will present in French. My slides are in English, but feel free to ask questions on my part in English or in French at the end. Donc, je vais présenter sur les revues de littérature qui ont été faites à l'INSPQ sur l'utilité et les conditions de faisabilité de la surveillance du SARS-CoV-2 dans les eaux usées. Une première revue de littérature qui est parue en février 2021 et une mise à jour qui est parue à la fin de l'année 2023. Next slide, please. Sorry. Donc, les objectifs d'apprentissage pour cette partie, on peut les sauter. Next slide. Parce que je vais présenter davantage la question de recherche qui avait été posée pour la revue de littérature et les objectifs d'apprentissage aussi pour cette revue. Donc, la question, c'était quelle est l'utilité et les conditions de faisabilité de la surveillance du SARS-CoV-2 dans les eaux usées d'une perspective de santé publique. Et les objectifs de la revue, c'est de documenter la validité et l'utilité de cette surveillance, d'explorer les préférences méthodologiques, c'est-à-dire les barrières et les conditions facilitatrices pour faire cette surveillance et d'informer au sujet des actions de santé publique qui peuvent être posées sur la base de ces données. Next slide, please. Donc, au niveau de la méthodologie qui a été prise pour les 2 revues de littérature, c'était une méthodologie de revue systématique de la littérature, revue de type systématisé, sans méta-analyse. Les bases de données pour la littérature blanche qui ont été révisées, il y en avait 4, Embase, Environment Complete, PubMed et Web of Science. Essentiellement, il y a 2 termes qui ont été utilisés avec les différentes variantes possibles en anglais et en français, des eaux usées et du virus SARS-CoV-2. Et la littérature grise a aussi été révisée, mais plus pour bonifier la discussion à ce niveau-là. Il y a des critères d'inclusion-exclusion qui ont été utilisés pour sélectionner les papiers. Donc, au niveau des critères d'inclusion, il y en avait plusieurs, mais parmi les plus importants, il y avait que les études se concentrent sur l'échantillonnage au niveau des usines de traitement des eaux usées. Donc, ça ne considérait pas là, les études où l'échantillonnage était plus en réseau ou au niveau des bâtiments. Et essentiellement, les usines aussi qui étaient implantées dans des villes de 150 000 personnes et plus, donc plutôt des grandes villes, comme suggéré là, par la Commission européenne pour l'implantation, la mise en œuvre là, de ce genre de surveillance en Europe. Et aussi, les études devaient faire un lien quantitatif là, entre les séries de données des eaux usées et les données cliniques, ce qui nous amenait à des papiers en général là, un peu plus grande qualité. Parmi les critères d'exclusion, il y avait toutes les études qui étaient davantage de la modélisation, puisque ce n'était pas parmi l'expertise là, de l'équipe de recherche. Également, les études pour lesquelles il manquait de l'information au niveau de la méthodologie de l'étude et les études qui focusaient essentiellement sur les aspects d'analyse en laboratoire ou sur les variants, là, n'étaient pas des études qui étaient incluses, elles étaient exclues. Next slide. Donc, il y a 35 études qui ont été retenues et 4 revues, et il y a une évaluation de la qualité qui a été faite là, pour ces études-là. Il y a une grille qui a été bâtie là, lors de la première revue, une grille d'évaluation de la qualité qui a été bâtie parce qu'il n'existait pas dans la littérature là, de grille adéquate sur cette thématique-là. Et cette grille-là a été revue là, par un expert externe. Donc, c'était une grille de 11 questions avec des critères pour chaque question. Puis, il y avait un score de 1 qui était donné là, pour si tous les critères étaient répondus pour une question, il y avait un score de 1 qui était attribué. Puis, par exemple, les questions touchaient des domaines comme est-ce que la question de recherche était claire et bien justifiée? Ensuite, est-ce que la méthodologie de l'étude était claire, était en lien aussi avec les objectifs? Est-ce qu'il y avait une bonne description de la méthodologie qui a été employée pour l'échantillonnage des eaux usées? Est-ce qu'il y avait une description adéquate, aussi une bonne présentation des données épidémiologiques qui étaient utilisées pour faire le lien avec les données des eaux usées? Est-ce que les résultats étaient bien compréhensibles et en lien avec les objectifs de l'étude et leurs conclusions? Puis est-ce qu'il y avait une valeur ajoutée de l'étude d'un point de vue de décision de santé publique? Chaque étude était classée comme ça avec un score de qualité haut, moyen ou faible. Next slide. Ensuite, il y a des constats qui ont été faits sur la base du contenu de ces études-là. Puis, les constats aussi, là, ont été, il y a des niveaux de support qui ont été attribués aux constats selon le nombre d'études qui rapportaient ces constats-là et la qualité de chacune de ces études-là. Donc, il y a eu 9 constats là, avec un niveau de support élevé qui ont été faits, 10 avec un niveau de support modéré et 1 avec un niveau de support faible. Next slide. Donc, je vais présenter pour débuter les constats avec un niveau de support élevé en lien avec la validité et l'utilité de la surveillance des eaux usées. Donc, je vais vous dire les constats en rafale avec un peu une explication là, pour chacun. La surveillance des eaux usées permet de dégager des tendances là, de circulation de la COVID-19 dans la population à différentes échelles géographiques. Il y a aussi un potentiel d'utilisation de la détection de la COVID-19 dans les eaux usées comme un outil de détection précoce pour l'épidémiologie circulante là, de la COVID-19 dans les eaux usées. Ces données-là sont des données qui sont indépendantes des données cliniques, qui sont complémentaires aux données cliniques et probablement aussi que c'est une approche coût-efficace pour la surveillance là, basée sur les données cliniques. Next slide. Donc, les constats avec un niveau de support élevé pour les préférences méthodologiques. La standardisation qui utilise des bioindicateurs viraux comme par exemple le virus de la marbrure du poivron ou le crossphage donne pas nécessairement, rehausse pas nécessairement la corrélation avec les données épidémiologiques. Tout ce qui est processus d'assurance qualité interne et contrôle qualité pour les analyses en laboratoire des eaux usées doivent être priorisés. Il y a un impact là, de la matrice choisie des eaux usées, donc soit l'eau, soit les boues, et de la composition physico-chimique là, de cette matrice-là. Il y a aussi un impact de la grosseur, de la longueur et du type là, de système d'eau usée. Donc, est-ce un réseau unitaire ou un réseau séparé, là, les eaux de pluie et les autres eaux. Donc, il y a un impact de tous ces facteurs-là sur les concentrations de SARS-CoV-2 qui sont retrouvées dans les eaux usées. Il y a une tendance à une utilisation de plus en plus des données d'incidence là, pour ce qui est des données cliniques par rapport aux données de prévalence quand on choisit des données cliniques pour comparer avec les eaux usées. Ensuite, les moyennes mobiles hebdomadaires ou une autre méthode là, de lissage des données d'incidence, leur adoption réduit la variabilité et augmente les corrélations avec les données des eaux usées. Next slide. Dernière diapositive sur les constats avec un haut niveau de support en lien avec les actions de santé publique. Donc, il n'y a pas vraiment d'intervention ou d'actions de santé publique bien établies là, qui sont basées uniquement sur les données des eaux usées. Il n'y a pas non plus de niveau de seuil pour les concentrations de SARS-CoV-2 dans les eaux usées qui mènent là, directement à telle ou telle intervention. Next slide. Pour les constats avec un niveau de support moyen au sujet des préférences méthodologiques, donc c'est préférable d'inclure autant que possible un échantillonnage à haute fréquence, comme 7 ou 5 jours, par exemple. C'est sûr que ce choix-là doit être ajusté là, à la situation épidémiologique. Est-ce qu'on est en hausse, en plateau ou en baisse, par exemple? Est-ce qu'on est en forte circulation ou en faible circulation? Ça doit être également ajusté aux objectifs de santé publique qu'on poursuit avec cette surveillance-là. L'échantillonnage, 24 heures composite au niveau des eaux usées, donne des concentrations qui sont représentatives de ce qui se trouve là, réellement dans les eaux usées. La standardisation qu'utilise le débit peut réduire la variabilité des données des eaux usées. Donc, il y a une certaine variabilité aussi et des incertitudes associées avec l'expression virale fécale là, de ce virus-là dans les selles et ça amène une variabilité dans les données des eaux usées. Et finalement, l'échantillonnage, le transport et les méthodes d'analyse en laboratoire là, peuvent tout influencer les concentrations qui sont retrouvées dans les eaux usées. Next slide. Encore au sujet des préférences méthodologiques, il y a des défis en lien avec les données épidémiologiques qu'on choisit pour comparer avec les eaux usées, parce qu'il y a eu de la variabilité dans les stratégies, les pratiques et les capacités diagnostiques, là, autant par PCR que par dépistage rapide pour le SARS-CoV-2 au cours du temps et dans l'espace également. Les résultats des comparaisons varient, des comparaisons entre les eaux usées et les données cliniques varient selon les dates qu'on choisit, donc la période de temps qu'on choisit, puis également en fonction de l'espace qu'on couvre là, sur le territoire. La force des associations en lien entre les eaux usées et les données cliniques varie. Ça reflète de l'incertitude dans les données, mais aussi l'utilisation en général de méthodes statistiques qui sont plutôt simples et l'agrégation des données pour un ensemble d'usines d'épuration et de villes, ça peut aider à obtenir des meilleures corrélations entre les données cliniques et les données des eaux usées. Next slide. Au sujet des actions de santé publique, la revue a confirmé qu'il y avait une utilité pour confirmer les tendances temporelles et spatiales qui sont observées dans la population pour la circulation du virus et également pour faire une certaine évaluation de l'efficacité des mesures de prévention qui sont en place. Next slide. Il y a eu un seul constat là, qui a été identifié avec un faible niveau de support, c'était au niveau des préférences méthodologiques et ça rapportait qu'au niveau statistique, les corrélations semblent entre les données des eaux usées et les données cliniques semblent spécifiques là, à chaque station d'épuration. Donc, ça veut dire que les corrélations changent là, selon les données qu'on choisit selon la station. Next slide. Finalement, il y a quelques perspectives qui ont été dégagées de la revue, qui avaient une nécessité là, de faire des améliorations ou d'acquérir davantage de données ou d'expériences au niveau de l'influence des facteurs environnementaux sur les concentrations qui sont retrouvées dans les eaux usées. Il y a également une certaine variabilité au niveau de l'excrétion fécale qui doit être mieux comprise. Ensuite, une harmonisation au niveau des méthodes de laboratoire entre les laboratoires là, qui est nécessaire. Également, une réduction autant que possible des délais opérationnels pour permettre d'avoir des données dans les eaux usées en temps opportun pour la décision. Les méthodes statistiques qui sont choisies pour analyser l'association entre les données cliniques et les données des eaux usées doivent être affinées. Idéalement, également, des seuils d'action pour la santé publique pourraient être dérivés des seuils d'action de concentration virale dans les eaux usées. La collaboration doit encore le raffiner entre le secteur de la santé publique et du génie environnemental, qui sont les deux principaux secteurs là, concernés par cette surveillance. Et les méthodes pour détecter les variants dans les eaux usées et également les autres pathogènes là, doivent être affinées. Ma collègue Inès pourra en parler. Donc, je vais céder la parole à Christine qui va nous parler de l'évaluation qu'on a faite du programme québécois. Next slide.
Oui, merci Caroline. Donc, so yeah, I will then continue with the program evaluation that was done with the data that was gathered during the first year of the program. Next slide. So the learning objective for this portion is to learn about the program evaluation, of course. So we studied the capacity of the wastewater signal to characterize the transmission of the virus under different attributes. So those attributes for the validity of the signal, its representativity, its exhaustivity, and the timeliness of the data. And also the evaluation focused on the implementation of the program from the user perspective. So next slide. I will start with the validity evaluation. Basically, we try to answer the question, does the signal of SARS-CoV-2 in wastewater allows for epidemiological surveillance, meaning tracking the trend in real time? And second, we evaluate if it allows for an early detection signal. So what was done is the wastewater signal was compared to clinical data. We used the new cases reported, the number of tests, the hospitalization, and the positivity percentage. We perform visual analysis of the graph, and also we perform cross-correlation function. Next slide. Here are the results for the visual analysis. So in blue, in all the graphs, it is the same line, which is the wastewater viral load. This is the data for the provincial aggregated signal. So all the site that was tested at that moment. And then in orange, in all the graph, you have different clinical data. So on the upper portion and left, it is the new reported case. Left down is the number of tests. Up right, it is the hospitalization, and down the positivity percentage. And the dark black line indicates the changes that occur in the screening policies. Next slide. We see that there is a variable synchronization depending on the period and the clinical indicator chosen. The synchronization appears to be better before October 2022. And after that, so just before the second change in the screening policy, and after that, it seems to persist more for the lower graph, so the one for the hospitalization and the positivity percentage. So this is for the epidemiological surveillance. And for the early detection, it is not crystal clear that there are any advancement between the clinical data and the wastewater signal. It may sometimes be in advance, like for the the lower graphs, it's more in advance for hospitalization and positivity percentage at the end of the series, but we do not see the same result for all indicators and periods. Next slide, please. Then we went deeper in the analysis with the cross-correlation function. To perform this analysis, we used the clinical case only, and we did the analysis in 2 steps. So first with all the series for the clinical case, next slide. And then we split the data set to use only the data where we seen the visual synchronization. So next slide, this portion over here. So before we lose the synchronization visually. Next slide. So cross-correlation function allows to reveal any lag that may exist between the data set. So in the cross-correlation graph, the x-axis evaluate the lag in days, and the y-axis evaluate the level of correlation between the dataset for a defined lag. What we see in those results is that depending on the period chosen for analysis, we have a different outcome of correlation. If the full period is taken, the clinical case tends to outpace the viral load. But if we take only the portion where we have the visual synchronization, then the viral load tends to outpace the clinical cases and with a positive correlation. Next slide. Yeah, thank you. So this is what I just said. So basically, it's hard to get defined conclusion from these data because the lack of gold standard makes it difficult to have data that remains stable throughout the full period and to gather a strong conclusion. It would be very interesting to perform the same analysis with the hospitalization and positivity percentage, where we saw more synchronization throughout all of the period. Next slide. The representativity was also evaluated. So this evaluation focused on understanding what the viral load wastewater signal was composed of and how those variables may impact the signal. So to do so, scientific and grid literature was reviewed for the 3 main components of the signal, which are the SARS-CoV-2 concentration in number of copies of the gene N1, the water flow, and the population. Next slide. I will not go in details for all the variable because of the limited time, but in the end, what we got to conclude is that there are a lot of independent factors that we cannot control that can bias the viral load signal or create measurement error, and effort should be put in place to get a more precise understanding of all of those factors. So this is in line with what Caroline mentioned in their literature reviews. Next slide. Next attribute evaluated was the exhaustivity, where we tried to characterize how complete the data set was, and we characterized the missing data and the impact of those missing data. Next slide. During the first year of the program, we had 3 major sampling regimens, which were 7 days per week, 5 days per week, and 3 days per week. Naturally, we see that the planned missing data correlate with the sampling regimen, but accidental missing data somewhat is similar between all regimens. So we do not have more accidental missing data, whether it is a full regimen or a partial regimen. The accidental missing data were caused by whether lack of sampling by the treatment plan, delivery issue, or it could be also failed quality control in the lab. And also we had some days where we did not have the water flow, which impede the normalization process. Next slide. Yeah, thank you. The higher number of missing data for the planned missing data for the 5 and 3 days sampling plan made the data imputation more complex, and the daily indicator for those sites was just to be irrelevant, but a weekly indicator may be more relevant for a sampling frequency like those. Next slide, please. The timeliness was evaluated by comparing the logistic delay to get the data from the wastewater, and those delays were compared to delay to get the data from the new reported clinical case. Next slide. Here we have the delay comparison between both. So I will just explain the colors because it's hard to see. In green is the incubation delay. So for the clinical, is the length of time between infection and symptom. And for wastewater is the length of time between infection and fecal excretion. So slightly shorter for wastewater. Then in blue is the delay related to sampling, orange transport, yellow transport, and orange the lab analysis. And the purple is the data dissemination. So overall, we see that clinical data takes 8 days, while wastewater takes 13 days. But the real major difference is the purple step, which is the data dissemination, which takes way longer for the wastewater compared to the clinical data. And that was mainly caused by the fact that wastewater data were published only once weekly, while clinical data were published daily. Next slide. Last portion of the evaluation was the appropriation of the program. So the question we tried to answer was how has been perceived the implementation of the program from the perspective of the user. This was evaluated by a survey sent to the user, and we had question with the scale of scale 1 disagree and 10 agree. And we are also open question, and different sub-attributes were evaluated as listed on the slide. Next slide. A lot of results came out of this survey, and I show you here 2 major graphs that came out of this portion of the evaluation. So on the left, it's a graph for the acceptability of the program. The blue line are in agreement and orange disagree. So a huge majority of the user agreed to the program definition, implementation, the indicator development, and its use to follow trending. But the fourth section, it is more medicated, and it's used for rapid responsiveness. And this totally makes sense with the previous result I showed you with the long delay for data dissemination. On the right are data for the user habilitation, where we add mixed responses for all the categories. So basically, user need more support to have a better interpretation of the indicator, more support to be able to properly communicate the data, and also to use it as a tool for surveillance. Next slide. So in the end, many recommendation comes out of the evaluation. Some of the main findings are listed here on the slide. So like diminishing the data dissemination delay to make it more useful for rapid responsiveness, which is something we had worked on with where we put the data on the online platform where user can perform customizable queries. So it would be easier to disseminate the data more quickly. Also, it would be important to prioritize 7-day sampling frequency if we want to have a daily indicator. If not, it would be relevant to switch to a weekly indicator for a lower regimen sampling. And also, other recommendation would be to develop knowledge transfer tool to help the user understand the data, implement mechanism for ongoing feedback with the user to understand their need as well. And also, one last recommendation would be to prioritize different pathogens of interest according to the epidemiological situation and plan implementation accordingly. So that sums it up for the evaluation. Ines, it's your turn.
Thank you. So last but not least, I'm going to talk today about how we are monitoring SARS-CoV-2 circulation and diversity through the community wastewater sequencing in Quebec. So next slide. So as a big objective, just to keep in mind that wastewater surveillance for SARS-CoV-2 variants is crucial also in monitoring the presence of the virus in the population and potentially predicting future outbreaks. So by analyzing the viral load in the wastewater, as we just saw, we gain valuable insight into the community infection rates and potentially the effectiveness of control measures what they are put in place. But furthermore, it allows us to identify the diversity of variants circulating within the population. And then tracking epidemiological trends of SARS-CoV-2 and its variants can enable and from public health intervention to mitigate transmission if needed. Next slide. So to keep in mind regarding the SARS-CoV-2 variants, it's that they are categorized based on genetic changes that occur during viral replication. So these changes are driven by mutations or viral recombination and lead to continuous evolution of the virus. You can see on the slide the genome of the SARS-CoV-2 virus. And so what we observed through the pandemic is that SARS-CoV-2 has consistently mutated, giving rise to variants distinct from the original viruses. So during the presentation, I'm going to use two terms to say the same thing. Actually, it's variant or lineage. It's basically subtype of the virus that are different to other virus by accumulation of different mutation. And why are we interested in this mutation and in variant in general? It's because this mutation can confer different advantage to the virus, either by increasing the transmission or the escape to the immune system or resistance to some kind of treatment. So it's because of that we're really interested in variants. Next slide. And so the way we are monitoring right now the SARS-CoV-2 variant in Quebec is was first by the sequencing, the representative sequencing of clinical samples, which was really well put. I mean, we sequenced a lot of samples in the beginning of the pandemic, and it helped us a lot to follow the circulation of the different variants, but unfortunately it has its limitations because we know, and has been shown previously in the presentation, that we underestimate the cases number, especially right now when people don't have all people don't have access to the PCR testing like it was at the beginning of the pandemics. And also our variant detection through this representative sequencing is really dependent on the screening strategy. Next. And for that, we wanted to have a look at the more like community scale screening of this variant. For instance, even when everybody had access to the PCR screening, asymptomatic person infected but not showing symptoms wouldn't even have the test because they were asymptomatic most of the time. So, but if you want to do a community scale screenings, next, this is where you want to go to see how it goes for the SARS-CoV-2 in the wastewater, because then you have access to all the community. Next slide. So quickly on the methodology, it was presented previously. So we are actually using the same samples that Christine just presented. And right now it's mainly 2 sites, as she said, 3 days per week. So the 2 sites are Montreal and Quebec. And so at the beginning of the week, there is this collection of the samples. They are sent to the different labs where they do the filtration, RNA extraction, and then the PCR. But also we do, for sequencing, an RT-PCR, an amplification of the whole genome, a preparation of the library. So this is how you prepare the genetic material to go to the sequencer. So we are using Illumina sequencing technology. It takes approximately 4 to 3 days to do all these steps. Next. And then after that, you have the bioinformatics analysis. I won't go through the details regarding these different steps, but just to keep in mind that it was a pretty complicated steps on our side because we wanted to be sure that what we are sequencing is actually what's there. So because of that, you have a lot of quality control steps and make sure that the variant that we get at the end, the mutation identified and the lineage characterized are the right one. So because of that, there's a lot of different steps, but basically, you do the quality filtering, you remove for instance, the human genetic material, the human reads. We use the same kind of method and from the clinical sample, we use the SARS-CoV-2 reference genome, we align to the genome, and then we can identify the different mutation comparing to this reference. And after that, we can identify the different lineages. But what is really interesting, I think, for you and actually for our fellow, for our collaborator epidemiologist, is this table that you can see here. Is the lineages frequency table. This is really the output we are generating. And for instance, in this table, there's a lot of information, but to be short, there is, for instance, 4 samples in this one. If you look at the column cities, you can see that's 2 samples from Gatineau, 1 from Sherbrooke, and 1 from Montreal. It was at the time when we were sampling other cities. And you have like different lineages and their relative abundance for each sample. Also, the one information that is really important for us when we do the sequencing is the coverage. So the coverage, what we call the coverage, is actually the percentage of genomes we were able to retrieve through the sequencing. And as you can see, for instance, the first 4 lines, they are the same samples, so 4 different lineages, and you can see the relative abundance, but also the coverage, and you can see that even if we retrieve only 35% of the genomes, we are still able sometimes to identify lineages if they are the right portion of the genome where the mutation are present. Next slide. And so once we send that to our colleagues at InfoCentre, so the epidemiologists, they can actually represent the data on this way. So each column is actually a CDC week. So it's an example of wastewater results, sequencing results that we did in 2022. So the CDC weeks, we can see that it's like the end of the summer through October, through the fall. Each week we represented it with the different proportion of lineages observed. As you can see on the right, the different lineages that were circulated at the time. So if you remember, there are the Omicron sublineages, BA2, BA4, 5, and their sublineages. And we did a comparison. This is what I think would interest you here, is the comparison of these results, sequencing results from the wastewater to the clinical sequencing that we've done. At the time, we were sequencing about between 500 and 600 samples per week in order to identify the lineages that were circulating. And we did the comparison like week by week between the wastewater and the clinical samples. And as you can see on these figures, the trends are pretty similar. It was really consistent results between the 2 methods with some slight differences. For instance, if you look at one sublineages of BA4, the orange one, the dark orange on the clinical samples, it was in higher proportion that was detected in the wastewater. And the reason of that is that we noticed that from some sublineages, the wastewater sequencing was performing not as well as the clinical to identify some of the sublineages. And one question that we were asked was, can the wastewater sequencing method allow us to an earlier detection of some variant before we see them in the clinical samples? The answer is it depends. For instance, if you look at the end of the period in October, this is the period of time where the new lineage BQ1 started to rise, and actually the number of cases raised with it, and it became the main lineages during the winter. And thanks to with the wastewater, we were able to detect it 2 weeks in advance comparing to the clinical. But if you look at the end of the summer, when BA4 and BA5 started to rise comparing to BA2, then they were detected earlier in the clinical samples. So it's actually depends. Next. And why we can have sometimes differences between the wastewater and the clinical sequencing. It's because of some limitation that goes with the methods, particularly in times where we have, like right now, low viral load, so not a lot of circulation of the virus in the province. So the level of virus in the wastewater is really going down, and then it's getting more difficult to detect the virus and to sequence it. And also because, as I said, sometimes you have part of the genome that are not well retrieved, and we did some analysis on the degradation level depending on the gene of the genome, and we could see that actually the one that is used for the PCR, the N gene, is less degraded. So you can see that this is the one with the red arrow, comparing to the first part of the genome of 1A and B and the spike, which are the main genes where you can see a lot of mutations that are selected and then used to identify variants, and these ones are more degraded. So because of that, unfortunately, as you can see on the right, so there are more recent results with more recent lineages, and then you can see more inconsistencies between even 1 week to another, because it's getting more complicated to identify variant. And sometimes, like you can see the gray column, it means that we didn't detect anything at all. The sequencing just didn't work because of this limitation. Next. And another limitation that we have using just only the method identifying lineage is that sometimes it's not that informative, even at the clinical level. Why? Is because since 2022, we see a huge convergence in the evolution of the virus, meaning that you have like different lineages acquiring the same mutation because they are conferring advantage to the virus. So this mutation, they are the ones that are of interest to us and not the lineage, because at the end you have like you can see in these figures that seems really complicated. It was an example of all the different sublineages we had at the time and the different mutation characterizing them. And as you can see, there was this mutation, the R34016, the spike, that was like appearing independently in different lineage. And look, at this time, we weren't interested in these like 15 lineages with this mutation that were all rising at the same kind of rate, but we were more interested in the mutation. So because of that, we are now developing methods to just look at mutation and not only on lineages. I think it's something that's already done at the federal level. Next. So to conclude my part, what we saw during this period of time sequencing both clinical and wastewater is that the sequencing individual clinical samples yields high quality sequence data, still really useful, but it is dependent on the screening test. Whereas for the screening, for the sequencing from wastewater samples, it allows for the broader detection and characterization of the SARS-CoV-2 variants within the community, and we got good quality that was comparable to the clinical. But the main I think the main thing we can learn from the presentation, and it was said by Caroline and Christine, is that this method is really complementary to the sequencing of clinical samples. And on our side in the lab, the future development we are working on are really doing the same thing, but for the airport wastewater, because if you remember, there were one time in the pandemics where we were sequencing a sample coming from the travelers at airport with the collaboration with the federal, but now it's not done anymore. So we are now looking to see if we can detect new variant coming from other countries before they start to circulate in the population within the airport. So I thank you for your attention. Next, and I want to thank for sure it's not only our work, the 3 persons who presented today, it's the work of a lot of people in our different teams, so we want to thank them, and I want to thank you for your attention. So if you have any question, you can go ahead in both languages for me too. Thanks.
Thank you very much to our presenters for this very rich and insightful presentation. Merci beaucoup. So we have a couple of minutes only left for the question period, and I wanted to direct your attention to the question and answer box. Our presenters have been very responsive in already answering questions. So we do have a couple of questions. At this point, maybe I'll open the floor to our guests to let you answer the questions that maybe you wish to speak to, since we just have a couple of minutes left, or you would perhaps want to elaborate on some of the answers that you've given already.
So the first question is, how did you account for differences between wastewater treatment plants when combining them? And what correlation analysis did you use to compare clinical lineages to wastewater lineages?
For the first question, I can answer for this one. So yes, we've seen difference when the signal was aggregated or taken individually between each treatment plant. For the early detection signal, it seemed to be better when taken individual treatment plants. So the aggregation of the signal may dilute the correlation at that point. But it was again variable for indicator and chosen and the period that was taken for the analysis.
And for the correlation analysis, unfortunately, for the lineages, unfortunately, I'm not the person who did them, so I cannot answer the question. I think, Caroline, you are answering the second one.
Yes. Okay. So I'm saying that for the moment, unfortunately, we don't have any really any methods to take into account the individual the variability in the fecal excretion. So we can inform, we can take information in the literature about that, but in our analysis, we are not taking this into account for the moment. I don't know if there is other question.
It looks like all of the questions in the Q&A box have been answered. I was actually curious in terms of from a health equity lens, and I'm thinking of like northern and remote communities. Could you speak to some of the perhaps benefits and challenges of wastewater surveillance when it comes to health equity?
Maybe I can say that for the pilot project, so the project that was before the program, we try to do some surveillance in remote region. So it was not necessarily indigenous community, but it was for sure a rural region with some really small cities. And yeah, it was difficult to convince all the small municipalities to get into the project. So there is really, there are many of them. So it's a lot of people to convince. And it was also at the beginning, so this method was not really, well known. And also there is some sampling and transport difficulties, challenge. Because they are removed, so it can take so long to get the laboratory and sample can get degraded. So Maybe we have to think about methods to analyze the sample on site. So that's a thing that they are doing in the north, in Quebec, so in the Nunavik, and yeah, so they want to develop method to analyze on site, and also for the sampling in the Nunavik particularly, there is some different challenge because there is no réseau, comment on dit réseau?
Network. There are no sewage network.
Yeah, so it's a truck that is going from communities to get the wastewater. So they have to think about a method to sample the wastewater from the truck. So there is some different there is some particularities and there is some challenge. Yeah.
Perfect. Thank you so much for your responses. This was really insightful. I'll just pass it over to Signe to close us off. Thank you.
All right, so we are nearing the end of today's seminar, and we would like to take this time to thank our speakers for sharing their insights and expertise. And thank you all of you for joining us today for the 6th seminar of Surveillance Advances. We hope you enjoyed this seminar. And I would also like to note that NCCID has a number of projects related to wastewater surveillance, and you may be interested in finding those on our website. Please take some time to complete our survey for this presentation. You can scan the QR code on this slide to access the survey, or look for the link in an email that will be shared shortly. Your feedback is important to help us identify areas of success and improvement for this series. You will also be able to request a letter of participation near the end of the survey. A reminder that the recording for today's seminar and the presentation material will be accessible for viewing on NCCID's website in a few weeks. If you have more questions about the Surveillance Advances seminar series, please visit NCCID's website at nccid.ca. Our next seminar will be held on Tuesday, April 30th, 2024, and this concludes today's seminar. Thank you all for joining us, and see you next time.